Hello, I'm Chong and I'm a postdoc associate from the Department of Veterinary Population Medicine. I'm excited to share our ongoing research today on swine influenza resultants in pigs from the field. But before that, let's talk about swine influenza virus. Swine influenza virus infection is a costly disease for swine industry and also poses a threat for human public health. The virus can be divided by subtypes and genotypes according to their genome, which consists of its RNA segments. Among them, there are two RNA segments are coding for HA and NA proteins, and they both together determine the virus subtype. However, the virus genotype depends on the whole set of 8 segments. Based on the combination of the types of each of 8 RNA segments, the influenza viruses are categorized into different genotypes. The segmented genome enables two different influenza viruses to exchange their gene segments with each other and produce the progeny virus with totally different genotypes. This process called resortment, and the produced new progeny viruses are also known as the resortants. Compared with the parental viruses, the resortants have different genotypes but may still share the same subtype. The influenza resortment greatly expands the virus genetic diversity and facilitates the co-circulation of different kinds of viruses in swine farms. Even though vaccination is widely applied in the US farms, however, the circulated strains are evolved much faster and it's hard for a single vaccine to have a perfect match to all the different circulating strains. As a result, some prevalent viruses may escape the immune pressure and finally cause the breakthrough infection in vaccinated pigs. Therefore, reducing the influenza diversity in pigs should be a key strategy for influenza control. To achieve this goal, we should first figure out how often and how badly does the influenza resortment happen at the individual pigs in the farm, and whether some resortants are more easily propagated on swine and human respiratory tract than others. And these are the knowledge gaps we address in this project. In our project, we took the nasal swabs of individual pigs from the swine farm that are experiencing the multi-subtype influenza co-circulation. Then we performed the experiment called plague assay to separate the individual virion clones from the virus population in a sample and culture them individually. We submit all the isolated influenza clones for whole genome sequencing and assign them genotypes through bioinformatic analysis based on how different each gene segment is from other virus clones. And finally, we will select the representative strains to test their growing abilities on the cells from swine and human respiratory tracts. Currently, we have isolated 243 influenza clones from 23 individual pigs, and these viral clones could be classified into 24 different kinds of viruses, which defined by genotype here. We found 8 pigs shed at least 2 different kinds of viruses at one time, and among these pigs, most pigs shed 2 or 3 kinds of viruses, and one pig shed as many as 10 different strains at sampling. Besides, we identified at least 2 different subtypes of the shed viruses from pig number 1 and 8. We believe this project will help us understand how influenza resortants emerge, persist, and subside in pig populations, and the result will also highlight the need to develop more targeted strategies to decrease the likelihood of resortment events happening in pigs and reduce the influenza infection in swine and human populations. Thank you for your attention.